Yes, good evening to all of you. My voice is audible. Please let me know. My voice is audible. Good evening to all of you. Please let me know. My voice is audible. Good evening to all. My voice is audible. Good evening to all. My voice is audible. Please let me know. Any of you? Okay. 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 So good evening to all. How are you all? Hope you are fine. Okay. How are you all? So how are you all? Everything is fine. My voice is audible. Okay, okay. So I have given you some homework also. Have you done it, all of you? The homework which is given to all of you, have you done it or not? Yes, please let me know. Okay. So now my voice is audible. All of you, please let me know. Okay, so I just asking you that I have give you some homework yesterday. Have you done it, all of you? Yes, good evening to all. Have you done the homework, all of you? Please let me know. Have you done the homework, all of you? Okay, okay, okay. Very good, very good. So we have to done supplementary genes. Then you have to write the interactions which are there, like dominant epistasis, recessive epistasis, duplicate genes, complementary genes, inhibitory genes, additive genes, along with the examples and along with their phenotypic ratio. So I hope all of you have done it, because I check some of your assignments. But I don't know your full name. So I request you, when you are submitting the assignment, please write your name also here, so that I will able to, like we have KN, but I don't know the names basically. So please write your name also if possible. When you are clicking the photos, just on the very first page, please write your name. So all of you have done it, but I am unable to tell the names of all of you. So yes, I know you have done it. If anyone left, so it's my request to all of you to please do the homework, as I am making the record of all these assignments which you are submitting to me. I am making the record how many students are attending the classes. The record of it is also there. So like we have 96 participants for this lecture series, but we have only 17 to 18 or 20 students at one moment. So I am making the record of everything. I am just taking your assignments also. The assignments are there, everything is there. And are you getting my voice? So are you getting my voice? My voice is clear to you? Yes, yes, good evening to all of you. My voice is clear. I think there is buffering right now. So my voice is clear. Any one of you, please let me know. Okay. So I just let you know that we have 96 members in this genetics class, but only few of you are attending it continuously. So everything is we are maintaining assignments also. So please try to submit the assignments on time, so that there will not be any, at the end when you want the certificate, there will not be any problem. So I know you have some exams, or you have some work also. So you can watch the sessions later also. But the only request which I have, you have to submit your assignments only. Getting my point? If you are unable to join the class right now, no issues at all. In the class because of some reason, because of exams, because of papers, no issues at all. But at least all of you have to submit the assignment. When there is some homework, you have to submit the assignment as soon as possible. Okay, so that is the only thing which I want to share with all of you. So are you getting it, or you are not getting it, what I want to say basically? Okay, so thank you, Archana. Thank you all. There is only a network issue. I just charge the phone right now. No issues at all. It will be okay soon. Don't worry. So yes, let me show you the syllabus now. So we have done Mendel's law of inheritance. We have done types of gene in action, and also we have done quantitative and qualitative characters yesterday. So today we will move forward with multiple alleles. So first I will try to take multiple alleles, then we will move further. So this is the part which is left yesterday, which is multiple alleles. So I will try to complete this first, and later on, because multiple allele is a very simple and a short topic, so later on I will start the another thing which is there in your syllabus, which is not related to genetics. Basically it is related to cell organelles, related to the very basics of biology. Like we will study fine structure of gene, ultrastructure of cell and cell organelles, and then structure of chromosome, mitosis and meiosis. So these 8, 9, 10 and 11, these four topics, if you are able to see here, these four topics, they are quite related with each other. 8, 9, 10 and 11, these are very related to each other. So we will try to complete this now, because the topics are very simple. They are there, the topics are there from your bachelor's, but I will start from the very basic. So after these 1, 2 and 3, I will try to work on this fine structure of gene, structure of cell and cell organelles, chromosomes, mitosis and meiosis, because these four are related. After it we will start the DNA replication, a transcription, translation. Then we will move forward with these four topics, because these four topics are interrelated. Getting my point? So we will do the topics like that which are quite interrelated with each other. So we will continue it as such. Getting my point? So today we will try to do multiple alleles, and then we will start with the genes. So is it clear to all of you? So your topic is very easy and very simple. But yes, we have to keep it in very briefly and with very basic, so that all of you will able to understand genetics in detail. So shall we start now? Everything is clear to all of you. Shall we start now? I will share the PPTs which we have. I hope I shared it yesterday or not. If I am unable to share the videos, if I forget to share the videos, so you can just remind me at any time that ma'am, please share the videos in the share the PPTs in the group, so that all of you will be able to all of you will be able to read those topics when you are free. Okay, so I am not forcing any one of you to just attending the classes on time. If you are busy, no issues at all, you can watch it later also. But the only request which I have is to assignments, because your assignments are submitted and there is a proper record of your assignments. So it's my request also, when you are submitting the assignment, please put your name on the very first page, so that I will able to know that this is Samiksha, this is Archana, this is Nikita, this is Anusha, this is Shivani. So it's my request to all of you that please put your name on the very first page. So I think that's all. Now we have to start the session. We already wait for 10 to 12 minutes for the students, but they are not here. So it's okay. Now we will start now. So we have to discuss today multiple alleles. So what are basically the multiple alleles we have to study now? So multiple alleles, what are basically multiple alleles? What are the features of multiple alleles and the common examples of multiple alleles? The common examples of multiple allele is blood group and coat color in mice. As I already told you, when you are doing any type of interaction, when you are doing anything in genetics, you have to remember that particular example. Like in case of Mendel, the example is Pythium sativum, that is the pea plant. So in case of linkage, when we will study, that means in case of Morgan, it is fruit flies. So you have to keep remember the example. Similarly, in case of quantitative characters, which we have done yesterday, the example is skin color, or you can say eyes color. So you have to keep these things in mind. Now, what are basically multiple alleles? Multiple allele, when more than two different forms of a gene exist in a species. More than two different forms of a gene exist in a species, they are called as multiple alleles. Normally, individual has two alleles. During the study on genetic material, Mendel also assumed only two alleles of a character. Getting my point? Suppose for height, we have either tall, or we have either capital T, or we have either small t. So there are only two alleles for a particular character. Like for roundness also, or for the color also, it will be yellow or green. So only two traits are here. So there can be larger number of possible allelic forms in that same population. This situation is called multiple allelism. So instead of two alleles for a particular character, we have many allelic forms. Are you able to get me? Suppose we have either red flower or white flower. So these are two characters. Tallness, tall and dwarf. Capital T, small t. These are two allelic forms. Okay. Roundness and wrinkleness. It's another two allelic forms. Then color, either yellow or green. It is again the two allelic forms of a single character. Either tall or either dwarf. So this is only the thing which is there in case of normal individuals, which have only two alleles for a particular character. The character may be height, the character may be eyes color, The character may be flower color, anything. The character may be anything, but the character has only two allelic forms according to Mendel. But what happens? There can be a larger number of possible allelic forms in that same population. This situation is called multiple allelism. So when we have more than one allelic form, not one, more than two allelic form, then it is known as multiple allelism. Let me check it whether it is, are you getting it? Are you able to get it? Okay. I hope you will get it. Let me do it further. So we have some features of multiple alleles. So multiple alleles always occupy the same locus on the chromosome. Chromosomes which we have to discuss in detail. Multiple alleles always influence the same character. That means they will only for a single character. The character may be height, the character may be color, anything, but multiple alleles influence the same character. No crossing over among the member alleles of the same multiple allelic series. So no crossing over among the member alleles of the same multiple allelic series. The common examples of multiple allele is blood group in human, coat color in mice, coat color in rabbit, eye color in Drosophila. So only instead of two alleles we have multiple alleles. So we will discuss the example of blood group in human. So in humans, the most common example is in the blood group of human. So gene coding blood group has 3 allelic rather than 2. So we have either, as we all are aware of it, that for HBO gene there are 3 alleles, IA, IB or small I. So we have 3 allelic forms, IA, IB and small I. So which we typically call these as A blood group, B blood group or O blood group. So the letter I, it stands for immunoglobulin. So the IA and IB shows codominance. So this is the thing which we discussed on the very first day, that what is basically codominance, right? So what is basically codominance? So here IA and IB, IA and IB shows codominance. An individual who is heterozygous for these two alleles, the phenotypes of both alleles are completely expressed, thus producing the blood type A and B. So AB blood group is by the togetherness of IA and IB. So IA and IB are showing the codominance. The small I allele, which is specifically for the O blood group, this is recessive to both IA and IB. Getting my point? So if the genotype is capital IA, the phenotype, the physical appearance, or you can say the blood group will be A blood group. When the genotype is B, capital IB or capital IB small ib, it means that both these are dominant over small I. Both A and B, IA and IB, they are dominant over the small I. Otherwise what happen? Only one is dominant over the another. According to the Mendel's ratio, one is dominant, one is recessive. But here two are dominant, so that is why they are known as codominant, and they are dominant over the single which is small I. So when IA and IB will present together, the blood group will be AB. When small I present, the blood group will be O. So this is the case of blood group in humans, where the two alleles are able to express its own self, and the two alleles are codominant to each other, and they will have equal effect on the phenotype. After it we have coat color in mice. So the coat or the hair color in mice is determined by a single gene with a series of alleles. So the color is controlled by a single gene. The gene is single, but it has many allelic forms. So the alleles are basically brown, black, agouti, grey, albino and other colors of hair. So these alleles follow the order of dominance. Like agouti is dominant over black, which is dominant over albino. So when black is there, the chances of albino will be less. When agouti is here, the chances of black and albino is less. This means that agouti is dominant to black and albino. Black is dominant on albino. So we have only a single gene. We have only a single gene. Sometimes what happens, we have polygenes. That means many genes controlling a single character. But here single gene controlling a single character. But this single gene has a many allelic forms. So this is multiple allelism. More than two alternative allelic forms of a gene occupy the same loci in a pair of homologous chromosomes in the population are called multiple alleles. The determination of a trait by more than two alleles is called multiple allelism. So you have to keep in mind only one thing, that we have a single gene, we have a single character, but this single character, or you can say this single trait, it is controlled by more than two allelic forms. So this trait or character is controlled by more than two allelic forms. All the variants or alleles of a gene may be originated by the mutation of a single wild-type gene. So, why there are so many alleles of a single gene? Why there are so many variants of a single gene? It may be because of the mutation of that particular gene. So, multiple alleles occupy the same locus within the homologous chromosomes. It means only one member of the series is present in a given chromosome. Since only two chromosomes of each type are present in each diploid cell, only two genes of the multiple series are found in a cell and also in a given individual. Getting my point? Only two chromosomes of each type are present in a diploid cell. Only two genes are able to express itself. So only two genes of the multiple series are found in a cell and also in a given individual. The gametes contain only one chromosome of each type. Therefore, only one allelic form will be there. Also there are brown, black, agouti, so many colors are there. Only one allelic form will be representative in the phenotypic ratio. Also in the genotypic ratio also, we have so many of allelic forms, but in the single individual only one allelic form will be represented. So I will let you know everything regarding this chromosomes and allelic forms in case of chromosomes. When we will study chromosomes in detail, then you will get it also. So crossing over does not occur in the multiple alleles. Crossing over is a part in case of mitosis and meiosis, not in mitosis, basically in meiosis, which is there in your syllabus. So we will discuss crossing over there. So multiple alleles control the same character, but each of them is characterized by a different manifestation. When any two of the mutant multiple alleles are crossed, the phenotype is mutant type and not the wild type. So when we are crossing two mutant multiple alleles, the phenotype is definitely mutant type, not the wild type. So now the another example is multiple alleles. The example is in eye color in Drosophila. So what happened in Drosophila? We have 14 alleles. 14 alleles for eye color. So we don't have black or brown only. We have 14 types of color in case of eyes, which produce various shades from white to red. So we have various shades from white to red in case of eye color in case of Drosophila. So red eye color is the normal, or you can say this is a wild color. So it is the main color which is there. Red eye color is the normal, and it is dominant to others. But because slowly and slowly, because of mutations or something, the color gets changed. From red color it gets changes into white, coral, blood, cherry, apricot, eosine, buff, tinged, honey, ecru, pearl, every and white. So from red we are getting so many of the shades. So this is the cross which is there. You have to keep remembering the example. The very first is blood color in human, number 2 is coat color in mice, in which we have three types of colors, agouti, black and albino, and the last, the very important, is eye color in case of Drosophila. So instead of only 2 or 3 allelic forms, we have 14 allelic forms. So when we are crossing the red eye Drosophila with white eye Drosophila, instead of getting red eye or white eye, we are getting in between alleles. So we are getting the in between alleles. This is again the multiple allelism in case of blood groups. It was reported by Dr. Carl in 1900, who is known as father of blood groups. Presence of two types of proteins in the human blood. We all know antigens and antibodies which are present in the body. And this is a detection of A, B and O blood group. How we are able to detect the blood groups. So you have to keep the thing only in mind that both A and B, they are co-dominant to each other. They are able to express each other together. So they are co-dominant. This is again the antigen antibody reactions. And this is the cross of possible offspring if mother is A blood group, father is O blood group. What are the chances of the child? Then this is another factor or another term which is there, which is related to blood group. This is known as RH factor. Now what is basically RH factor is antigenic protein. RH factor is antigenic protein present on the surface of RBCs in human beings. So we have blood groups, we have A, B, AB and O blood group. Along with it, we have one more thing which is known as RH factor. So it was first discovered by Landsteiner and Wiener on rhesus monkey, on the RBCs of rhesus monkey. So also found in 85% of American and 93% of Indians, they are RH positive. Some persons are there which are RH negative. RH factor is controlled by a pair of genes, capital R and small r. There is some functioning of this RH factor which is related with the pregnancy in case of mother. There is incompatibility during pregnancy because of this RH negative. So when RH negative woman marries with the RH positive man, suppose the woman is negative, the father is positive, now the baby in the mother will definitely be negative with RH positive. So mother is RH negative with RH positive fetus. So baby inside here is positive. Mother is negative but the baby is positive. Or you can say RH positive. Why? Because from the father the baby gets positive RH. So cells from the RH positive fetus enter the woman's bloodstream. So now your body has negative, but from the baby the positive will come here. So woman becomes sensitized antibodies formed to fight RH positive blood cells. Because the woman body has negative cells and positive cells will come, the woman's body or the mother's body will fight definitely. Now what will happen? In the next RH positive pregnancy, maternal antibodies attack the fetal RBC cells. So not in the first pregnancy but in the second or in the third pregnancy, what will happen? The female or the mother's body will be prepared so much that the female's or mother's antibody will attack the fetus RBCs in the womb. So this is known as RH incompatibility. So there is not any problem with the blood group. The only problem is with the RH positive which is present on the RBCs. So you have to keep this thing in mind. This is again the example of human blood group. I already told you. So you have to go through it by your own self. Color blindness is not in your syllabus. I think that is the only thing which is there in the um, this is only the simple thing which is in case of multiple alleles. You have to keep in mind only about the basics in the multiple alleles. That instead of only two alleles, we have more than two alleles. Okay, like in case of Drosophila, instead of two alleles, we have 14 alleles. This is a very simple thing which we have discussed. The points are very simple, and I just keep it very basic. You have to remind the three examples which are there, and you have to keep in mind that instead of two alleles we have more than two alleles. Okay, so now we have another topic which we have to start now, because multiple allele is very simple. Now we have to start the cell and cell organelles. So please help me, have you done cell organelles before? Please help me if you have done it, any of you. I think all of you must have done it. The cell, what is basically cell, what are the organelles which are present in the cell. Have you done it before also? Any one of you, please let me know, so that we will cover this topic. And this topic is very important and very simple also. So I just want to cover it fast. So please let me know, have you done cell before also? Cell organ, okay, so you have done it. Okay, okay. Bhavni also done it. All of you, I request all of you, have you done cell? Because cell is very basic. It is from our plus 1 and plus 2 classes that we are able to understand what is cell, what are the organelles which are in the cell. It is not a part of, basically it's not a part of genetics, but till how it is there in your syllabus. So we have to complete it. Okay, so as you have done it, I will try to do it fast. And along with cell we have to understand chromosome. Along with it we have to study mitosis and meiosis. So definitely mitosis and meiosis will be there in your syllabus. uh, Other things we will discuss as we will move forward. I am just taking very simple topics so that you will be able to understand everything. Like some things are a little bit difficult for you. So that is why in between I am taking again the simple topics like mitosis, meiosis, they are very easy. Chromosomes are what are the structure of the chromosome, what is cell, what are the functions of cell organelles. So now I am just going to start, otherwise we will not able to complete it on time. So many things will be covered in this PPT, which I have made regarding cell and cell organelles. Okay, so let's start it now. Yeah. So we have 200 slides, 243 slides. I hope you all will cooperate, and you all will able to understand it. Because instead of only of cell, I have completed chromosomes also. Lots of things are there which I have put together. So are you getting me? Everything is fine. Okay. Okay, thank you all. Thank you, Abha, Aboli, Archana, and Anusha. All of you, thank you. So now we're just going to start it. So cell is a very fundamental unit of life. Yeah. So the cell is a very basic structural and functional unit of living organism. In other words, cells make up the living things and carry out activities that keep a living cell thing alive. So basically, cells, they are responsible for this whole, the living things which are present around us. So cell is responsible for all the metabolic activities, for all the structural and functional units of living organism. The cell is responsible for all those things. Now we have some theories which are associated with the cell, that how cell exists, how cell form, There are some scientists in the previous years, they have so many assumptions regarding the cell. So like these are the assumptions are all known living things are made up of one or more cells, right? So instead of one cell we are all the living things, they are made up of more than one or more cells. All living cells arise from pre-existing cells by division. So division means mitosis or meiosis. It's right. The cell is a basic unit of structure and function in all living organisms. So please understand this PPT well. I will ask you some questions which you have to do. So the cell is a basic unit of structure and function in all living organisms. So these three things are very true. So these are some scientists who are able to work on it. So in 1665 we have a scientist whose name is Robert Hooke. He was able to discover the cell very first. Getting my point? In 1883 we were able to isolate, or we were able to discover nucleus. So from 16th to 18th century, it takes so many long time to discover the cell organelle which is present inside the cell. Okay. So this is another scientist which in 1674 was able to observe the living cell. So this is the history basically. So then in 1835, so we have here 1883. In 1835, the scientist was able to discover fluid content of the cell. In 1838, the scientist was proposed that all plants are made up of cells. So slowly and slowly they are able to get completely about cell. So now in 1845, Carl proposed cell is the basic unit of life. So this is proposed by the scientist in 1845. Then in 1855, we have these two scientists proposed all cells arise from pre-existing cells. All cells they are arise from pre-existing cells. So these are some historical things which are there. Now we have unicellular organism which is made up of one type of a cell. We have multicellular organism which is made up of more than one type of a cell. I am not taking it, in that briefly because you have done it already. So I am just letting you know about simple things. We have unicellular cells organisms, we have multicellular organisms. In unicellular we have single cell. In case of multicellular we have more than one type of a cell. So this is the leaf cells, this is the muscle cells. These are the group of cells, or you can say the cells which are seen under the microscope. So these are the multicellular, that means more than one type of a cell. Now size of the cells. So cells vary in size, they are small, they may be large, the size may be different. This is the mycoplasma is the smallest cell. The ostrich egg is the largest cell. So the mycoplasma is the smallest cell and the ostrich egg is the largest egg. Is the largest egg, not only the largest egg, but the ostrich egg is the largest cell also. Then sperm cells, they are the smallest cell. Ovum cell is the largest cell. Nerve cell is the longest cell. Then these are the shape of cells. Cells vary in shape. Variation depends mainly upon the function of cells. Some cells like euglena, amoeba can change their shape, but most cells have a fixed shape. So they have the fixed shape. They either may be circular, may be round. Like RBCs are circular, biconcave for easy passage through human capillaries. So RBCs are circular, biconcave. Nerve cells are branched to the conduct impulse from one point to the another. So the nerve cells are basically branched. WBCs can change their shape to engulf the microorganisms that enter the body. So they have different shapes, they have different sizes, and we are able to see all these things under the microscope. Now these are the organelles which are there in case of animal cells. So the very first homework to all of you, please write it down anywhere. You have to draw the structure of animal cell. In the animal cell we have nucleus, We have Golgi body, we have vesicles, plasma membrane, mitochondria, cytoskeleton, centriole, lysosome, cytoplasm, rough endoplasmic reticulum, smooth endoplasmic reticulum and nucleus. So we have 12 cell organelles which are present in the cell. So when you will make the diagram, you have to show all these organelles. Getting my point? So when you will make the diagram of animal cell, you have to make all these organelles. So you have to label all these organelles. Like this is mitochondria, this is Golgi body, this is nucleus, this is nucleolus. So you have to draw everything. So this is your first homework. You have to draw only animal cell. If you want to draw plant cell also, it's your wish. Otherwise you have to draw the single cell along with its cell organelles. Now this is a bacterial cell. Bacterial cell consists of capsule, cell wall, plasma membrane, cytoplasm, flagellum, food granule, plasmid, ribosomes and nucleoids. So this is the bacterial cell. After it, the structure of a cell. Now what is basically the structure of cell? These things are there. When we are able to watch it under the microscope, we are able to get that many things. So if we study a cell under a microscope, we would come across three features in most every cell: plasma membrane, nucleus and cytoplasm. I will let you know also. So this is a plasma membrane, which is the outer membrane. Getting my point? So we have to start with the plasma membrane, nucleus, cytoplasm, and rest are the organelles. So these all are the organelles. Otherwise the main things are plasma membrane, nucleus and cytoplasm. Now plasma membrane is, all of you must aware, it's extremely delicate, thin, elastic, semi-permeable membrane. It is made up of two layers of lipid molecules in which protein molecules are floating. Thickness varies from 75 to 110. Can be observed under the electron microscope. So the plasma membrane is the outer membrane, which is basically two layers of lipid molecules. In between that lipid molecules, these protein molecules, they are floating. Then cell wall. Now cell wall is basically what? Cell wall is present in non-living and outermost covering of a cell, generally found in plants and bacteria. So cell wall is absent in case of animal cell. It is present in plants and bacteria only. It can be tough, rigid and sometimes flexible also. So it is made up of cellulose, hemicellulose and pectin. May be thin or thick. Okay. So the thickness varies from 50 to 1000. There are some functions of this cell wall. Now this is nucleus. The nucleus is basically what? It is a dense spherical body located near the center of the nucleus. The diameter varies from 10 to 25 micrometer. Present in all the cells except RBCs and seed cells. So nucleus is absent in case of RBCs. Well developed in plant and animal cells. Undeveloped in bacteria and blue-green algae. Most of the cells are uninucleated. Few types of cells have more than one nucleus. So this is a nucleus which is present inside the cell. If you are able to see here again, the nucleus has a double layer covering which is known as nuclear membrane. Like we have plasma membrane around the cell. Similarly, we have a nuclear membrane around the nucleus. So nuclear membrane has pores. So if you are able to see, we have the pores here about 80 to 100 nanometers. Their colorless dense sap present inside the nucleus, which is known as nucleoplasm. Like inside the cell we have cytoplasm. Similarly inside the nucleus we have nucleoplasm. So nucleoplasm contains round shaped nucleolus and network of chromatin fibers. Fibers are composed of DNA and histone protein. So inside the nucleus we have DNA and histone protein. So these fibers condense to form chromosomes during cell division. After nucleus we have after this nucleus we have cytoplasm. So this is again, the nucleus consists of chromosomes which consist of DNA. The chromosomes contain sets of DNA are called genes. Gene transfer the hereditary information from one generation to the next. So the functions are control all the cell activities like metabolism, protein synthesis, cell division. All these things are controlled by the DNA which is present inside the nucleus. The cytoplasm is the jelly-like material formed by 80% of water present between the plasma membrane and the nucleus. It contains a clear liquid portion called cytosol and various particles are also there. So you have to read these properties by your own self. Similarly, we have endoplasmic reticulum and we have Golgi body, which was discovered by Camillo Golgi, and it is formed by the sacs of 5 to 8 membranous sacs. Sacs are usually flattened and are called cisternae. So these sacs are known as cisternae. Has two ends. We have cis phase, which is situated near the endoplasmic reticulum, and we have trans phase situated near the cell membrane. So we have some functions of this Golgi bodies also, like it sorts and packs the material synthesized in the cell, delivers synthesized material to the various targets inside the cell, produce vacuoles and secretory vesicles, forms plasma membrane and lysosomes. So now this is your homework. The second homework is you have to let me know the functioning, you have to draw and you have to tell me about endoplasmic reticulum and Golgi body. I hope you will get it. So you have to tell me about endoplasmic reticulum, the type of endoplasmic reticulum, like smooth endoplasmic reticulum and rough endoplasmic reticulum, and the this Golgi apparatus also, this Golgi body. So we have this endoplasmic reticulum, we have smooth reticulum and rough endoplasmic reticulum. After it we have Golgi body. So you have to let me know about both of these. After it we have one more thing, which is mitochondria. You must know of this organelle, which is mitochondria, which is small rod-shaped organelle. It is bounded by two membranes. We have inner membrane and we have outer membrane. The outer membrane is smooth and encloses the contents of mitochondria. The inner membrane, so if this is the outer membrane, and the inner membrane is finger-like folded structure. So if you are able to see, we have finger-like folded structure, and in the form of cells, in these inward projections, they are known as cristae. So these finger-like structures are known as cristae. The inner cavity is filled with matrix, which contains many enzymes. So contain their own DNA, which are responsible for many enzymatic reactions. So this mitochondria is known as the powerhouse of the cell. Getting my point? So this mitochondria is known as the powerhouse of the cell because it provides us the ATP. So the ATP molecules are very much important for the activities of the living cell. So this is the mitochondria. Now this is plastids, which are present in case of plants. It may be either chromoplast, chloroplast or leucoplast. So these are again the double membrane bound organelles found inside plant and some algae. They are responsible for activities related to storing the food. So for food storage, plastids are quite important. They often contain different types of pigments that can change the color of the cell. So they are chromoplast, chloroplast and leucoplast. So chromoplast, like the color which is there in the carrots because of the pigment carotene. Similarly in the mango, the pigment is xanthophyll. In the tomato, the pigment is lycopene. So they all provide the color to the fruits. So then another important organelle we have is chloroplast, which is there in case of plants. Similar to the mitochondria, the chloroplast is also double membrane bound organelle, which is found in the plant cell. After it we have centrosome. So this is very important. We will discuss this centrosome in meiosis also. So it is a membrane bound organelle which is present near the nucleus. It consists of two structures called centrioles. Centrioles are hollow cylindrical structure made up of microtubules. I will discuss these things during the cell division. When we have to discuss mitosis and meiosis, that is tomorrow, we will discuss the centrosome also. Cytoskeleton is basically what? It is formed by the microtubules and microfilaments. They are hollow tubules made up of protein called tubulin. They only provide, what's the functioning of this cytoskeleton? It provides shape to the cell. So these are some of the organelles which are present inside the cell. we have some difference between prokaryotic cell and eukaryotic cell. Kindly read by your own self. Animal cell and plant cell. So this is the whole cell in which we have nucleus. Let me check whether you are here or not. So are you getting it till now? Okay, so you are here. Is it okay? Are you getting it or not? Because these are the simple cell organelles, that is why I am not taking so much long time on it. So this is basically the cell organelles. So this is the whole cell. In the cell we have nucleus, we have mitochondria, we have Golgi complex, we have ribosomes, we have vacuoles, we have lysosome, cytoplasm, everything is there, and each organelle has its own specific function. So you have to draw the structure of an animal cell, and you have to let me know about endoplasmic reticulum and the Golgi complex. So you have to let me about all those things also. Now this is a cell theory which was proposed in 1839. We have already discussed these points, that they are the cells that living things are made up of cells. Cells are smallest working unit of living things. All cells come from the pre-existing cells during the cell division. So we have all. Mummy दरवाजा भेज दो ना. मम्मी मम्मी. So all these, they are cells are made up of, all living things are made up of cells. They are the smallest. They are the smallest working unit of a living cell, and the cells, they are come from the pre-existing cells during the cell division. So these are again the constituents of which are present inside the cell. We have water, we have carbohydrates, lipids and proteins, along with some electrolytes also. So we have discussed about the cell membrane, which is known as the plasma membrane. It consists of a lipid bilayer along with some proteins which are embedded in between. Then we have cytoplasm, which is thick jelly-like substance, not substance but thick jelly-like fluid, which is found in both plant and animal cell, and this cytoplasm is responsible for holding all the organelles together. Then we have mitochondria, which is known as powerhouse of the cell. This is known as the powerhouse of the cell, and it consists of outer membrane and inner membrane, and they have finger-like projections which are known as cristae. So this mitochondria is responsible for many pathways which are there in our body, like citric acid cycle, respiratory chain oxidation. For all these things we have mitochondria. Similarly, we have endoplasmic reticulum. The functioning and the diagram we have to make it. We have two types of reticulum. We have smooth endoplasmic reticulum and we have rough endoplasmic reticulum. Then this is the Golgi body. So endoplasmic reticulum and Golgi body, they are quite related with each other. That is why I gave you this homework, so that you will go through these organelles in detail. Then we have cytoskeleton. We have discussed these organelles. This is the nucleus again, which is covered by the nuclear membrane, and we have pores in between which are known as nuclear pores. I will share this PPT. Kindly try to do it by your own self. Now this is the diagrams when we are watching it under the microscope, the electron microscope, how we are able to see all these things. So I will share the PPT so that you will be able to read these organelles in detail. This is again the organelles. They are just showing us the visualization when we are watching it under the microscope and how they will be looking now. So this is like endoplasmic reticulum. So when this is mitochondria when we are watching it under the electron microscope. So you have to go it by your own self and let me know. So this is whole eukaryotic cell when we are watching it under the microscope. So we have nucleus. So this large one is the nucleus. If you are able to see here, this foot shape is the mitochondria. Now these are the vesicles. Now this like that one is this one is the endoplasmic reticulum. The whole these one. So they are the endoplasmic reticulum. So just go through these diagrams. Again, this is the this is maybe the plant cell. So we have a very large nucleus. So cell wall is present. No chloroplast. Nucleus is large. So nucleus is quite large. So please go through these cell organelles, and yes, we have chromosomes also. So if you want to do it, we will do chromosomes today also. Otherwise, we will discuss it tomorrow. So you want to discuss it today or you want to discuss it tomorrow? Or cell organelles are sufficient for today? Please let me know. Cell organelles are sufficient for today. Which thing I have to repeat dear Komal? Let me please ask me if you have any query. I will share the PPT. If you will not be able to get it, then I will discuss it again also. Okay. So I will share the PPT up to organelles, and tomorrow we will start the chromosomes. Okay, okay. So it's sufficient for today. I think yes, we all, because it's not a simple thing. There are so many organelles, but I just want to tell you the very simple things which are there. The number of organelles. So you have to read it by your own self, the functioning of every organelle and everything regarding it. You have to read it by your own self. And what is your today's homework which you have to do? What is the homework which we have discussed right now? Any one of you, please write it so that all will be able, or either write it in a telegram. Otherwise, they will ask for the homework and homework again. So please keep this thing in mind that your assignments are with me and we are making proper record of it. Bhavna, what you are saying here, I am not getting it. Oh, I didn't send you any PPT. I think I sent you yesterday's PPT or not. Please let me know. I'm really sorry. I'm really sorry if I forget to share the PPTs. Maybe I just share in the MBPT batch and I just forget to share it in the group. So I will share it right after I have my another class, but after 10 p.m. I will share it in the group. Okay, so no worries. I will share it today, the organelles and the complementary genes and all the PPTs which are there. Yes, so I think that's all for today. So take care of yourself and we will meet tomorrow with chromosomes. In the meanwhile, you have to draw the animal cell structure and please keep it in mind. There should be everything, all the organelles should be there, and you have to make endoplasmic reticulum and the Golgi body also along with their functioning and along with how they are associated and everything. Okay. So that is all for today. We will meet tomorrow. Till then, yes, yes, please send your assignments to today or by tomorrow also because now it's 9. So don't worry, you can send it at any time, no issues at all. I think that's all for today. I just hope you are getting it. I'm sorry if I am little bit fast, but till take care of yourself. When you will go through the PPTs, you will definitely able to get it. If you have still any query, you can message me at any time on my personal telegram also. So that is all from my side. Take care. Bye bye. Good night. And I'm sorry I'm leaving because I have my another class. Meanwhile, I just request all of you to please share and subscribe my channel so that you will get the notification when I will get live. Okay. So that's all from my side. Please take care of yourself. That's all from my side. Take care of yourself. Goodbye. Good night. Just thank you all for because you are here continuously with me for the 1 hour long session. It is very difficult for all of you too also to continuously hear. I just want to thank all of you. So take care of yourself. Bye bye. Bye bye. Take care all.